Hello guys, welcome to our channel AskPrep. Today's series is for 11th class biology. We will be studying chapter 14 Respiration in plants in detail. We have also added important notes for your exam preparation. All the best for your exam. Starting with discovery and definition, respiration is a biological process in which oxygen is used to break down complex organic molecules into simpler compounds, releasing energy, carbon dioxide, and water as byproducts. This process is essential for all living organisms, including plants and animals, as it provides the energy needed for various cellular activities. The term respiration was first introduced by Henry Dutrochet. Cellular respiration is a crucial metabolic process involving the oxidation of carbon-carbon (C2C) bonds in complex molecules like glucose, leading to the release of energy in the form of adenosine triphosphate (ATP). This energy is used for various cellular functions. Respiratory intermediates and their roles. Various intermediates formed during respiration play critical roles in the biosynthesis of other important molecules. Acetyl coenzyme A is involved in the synthesis of fatty acids and gibberellic acid. Succinyl coenzyme A contributes to the formation of chlorophyll, phytochrome, and cytochrome. Oxaloacetic acid and alpha-ketogluteric acid are used to produce amino acids like aspartic acid and glutamic acid. Types of respiration based on substrates. Floating respiration, the most common form of respiration, in which fats or carbohydrates serve as substrates. Protoplasmic respiration occurs when a plant is in a state of starvation, and proteins are used as respiratory substrates. Types of respiration. Depending on whether oxygen is present during cellular respiration, there are two types of respiration. Anaerobic and aerobic respiration are the two types. Aerobic respiration, it happens in the cell's mitochondria when energy, or about 2870 kJ, is released. There are two primary phases of aerobic respiration, the citric acid cycle and glycolysis. Kolicker made the initial discovery of it in the striated muscles of insects. On the other hand, C. Benda coined the term mitochondria. Hobhoum postulated that the mitochondria serve as the site of cellular respiration. Mitochondria are referred to as the semi-autonomous organelles since they have their ribosomes, DNA, RNA, and proteins. They are found as endosymbionts, symbiotic relationships inside eukaryotic cells. Glycolysis. Glycolysis is the first stage of cellular respiration and can occur with or without oxygen. It breaks down one glucose molecule into two molecules of pyruvic acid, generating a small amount of ATP. The key steps include phosphorylation of glucose. Glucose is phosphorylated using ATP, converting it into glucose 6-phosphate. Formation of fructose 6-phosphate. The glucose 6-phosphate is isomerized into fructose 6-phosphate. Cleavage of fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is split into two molecules of triose phosphate. Oxidation and ATP formation. The triose phosphates are oxidized to produce ATP and pyruvic acid. See the image. Fermentation. Anaerobic respiration, also known as fermentation or zymosis, is the process that takes place in the absence of oxygen and involves the incomplete oxidation of food ingredients that produce CO2 and ethanol as end products. Yeast aids in the completion of the procedure. Schank was the one who came up with the name fermentation, while Gay-Lussac was the one who initially discovered the process. Fermentation takes place in a variety of organisms, such as many prokaryotes, unicellular eukaryotes, and seeds that germinate in an anaerobic environment. Alcoholic and lactic acid fermentation are the two different types of fermentation processes. Alcoholic fermentation, carbon dioxide and ethanol, ethyl alcohol, are the products of it. It takes two steps. First, glucose molecules undergo glycolysis, which yields pyruvic acid. Pyruvic acid is subsequently converted to carbon dioxide and ethanol. The two actions are as follows. An enzyme known as pyruvic acid decarboxylase aids in the conversion of pyruvic acid into carbon dioxide and acetaldehyde. The process of acetaldehyde, which yields ethanol and carbon dioxide, is aided by an enzyme known as alcohol dehydrogenase as well as a coenzyme. Tricarboxylic acid cycle. following the full oxidation of pyruvic acid, which happens in a sequence of step-by-step reactions and needs oxygen. This cycle involves the creation of carbon dioxide and water. This process occurs within the cell's mitochondria. Since Hans Krebs followed the steps, the cycle bears his name, the Krebs cycle. The cycle is also known as the citric acid cycle since its initial constituent is tricarboxylic acid, which is also known as citric acid. Since the cycle consists of 3 acids, it is also known as the tricarboxylic acid cycle. In the TCA cycle, acetyl coenzyme A serves as the respiratory substrate, and acetic acid, a 4-carbon complex, serves as the acceptor molecule. This cycle consists of 2 decarboxylation reactions, removing CO2, and 4 dehydrogenation reactions, removing hydrogen. Here, carbon dioxide will be produced as a result of the coenzyme reduction. Together with 4 carbon oxaloacetic acids, 1 molecule of acetyl coenzyme A is involved in the synthesis of citric acid, a 6-carbon chemical. Citrus synthase is an enzyme that helps this reaction happen. The creation of carboxylic acid is the result of this reaction, which needs a water molecule. Let's understand whole in detail.
With the help of a variegated leaf experiment, we can prove that chlorophyll is essential for photosynthesis. To do this experiment, we need a potted plant with variegated leaves. We have to keep the plant in dark for 3 days. Do you know why? So that all the starch present in the plant gets used up. Now place this plant in sunlight for about 6 hours. Now detach a leaf from the plant and mark the green areas of it. Now trace the leaf and its green borders on a butter paper. Next, place the leaf in boiling water for a few minutes. Take it out and place it in a beaker containing alcohol. Now place the beaker with alcohol into a water bath. Heat the setup till the alcohol begins to boil. The leaf loses its color. The alcohol turns into green color. Now take the leaf out and dip it in a dilute iodine solution for few minutes. Take out the leaf and rinse it. Observe the blue colored part of the leaf and compare it with the paper tracing. Here we observe that starch is prepared only in the green color parts of the leaf. Hence we prove that chlorophyll is important for photosynthesis. How can we prove that carbon dioxide is necessary for photosynthesis? Write an experiment to show that carbon dioxide is essential for photosynthesis. We can prove that carbon dioxide is necessary for photosynthesis by a simple experiment. To do this experiment, we need 2 healthy potted plants of same size. We have to keep them in a dark room for 3 days to detach them. Place the plants on glass plates or smooth finish tiles. Place a watch glass containing potassium hydroxide by the side of one of the plant. Cover both the plants with separate bell jars. Why to place the plants on glass plates? Because we want the plant to be in a airtight environment. So we have to take a smooth surface at the bottom and a bell jar at the top to keep it airtight. Even then some air can enter inside through the edges. How do we stop it? That is why we have to apply Vaseline or grease at the edges to prevent the entry of air. But still there is some air in the bell jar. That means there is some carbon dioxide inside it. How do we remove it? That is why we have placed a watch glass with potassium hydroxide, which will absorb the carbon dioxide present in the jar. So now our setup is ready. One plant without CO2 and another plant with some CO2. Let the plants stand in sunlight for 2 hours. Now pluck a leaf from each plant and test them for the presence of starch. Do both the leaves show the presence of same amount of starch? No. A plant with KOH plant plate shows no signs of starch, but a plant without potassium hydroxide could prepare some starch. By this experiment, we can conclude that carbon dioxide is essential for photosynthesis.
Thanks for watching the video. We will drop the notes in the description of this video. If you found this video helpful, please like, subscribe, and share. See you in the next video, and all the best for your exam.